All right, Anatomy 2 students. So this is the second half of chapter 25, the metabolism chapter. So in the first lecture, we went over the processes of breaking down glucose through glycolysis and the citric acid cycle and the electron transport chain. The rest of the chapter deals on how we use other molecules to either replace the glucose or to build glucose. Then a little bit later in the end chapter, we talk about different types of vitamins, fat-soluble vitamins, and water-soluble vitamins. So the rest of the chapter is a lot of vocabulary. Of course, I will ask you this vocabulary on the test, so you want to make sure either you learn the definitions or you can at least figure out the definition of each of these processes. For example, gluconeogenesis. Genesis means to make. Neo means new. And then glucose. So that means what we're going to be doing is we're going to be building new glucose from non-carbohydrate sources. So anytime we can string together 6 carbons, if we can get 2 3-carbon pieces, well, then we have something we can then put in through the process that we just talked about through glycolysis, mainly through the citric acid cycle. So gluconeogenesis is getting that 3-carbon source from any other molecule that's not pyruvate that's been broken down by glycolysis. Now, in our bodies, we store glucose in a long chain form known as glycogen, and it's formed in the liver, and it's stored in the liver and in the skeletal muscle. Glycogenesis is building glycogen. So we always build glycogen from excess glucose. So if you have a glucose-rich meal, your body takes in what it needs, and what's left, it takes to the liver, and it is made into glycogen. And so genesis and glycogenesis tells you you're making glycogen. Now, if your blood glucose drops and you need some glucose, well, then you need to break glycogen. And that process is called glycogenolysis. To lyse is to break. So what are we breaking? Glycogen. And that breaks it back down into its glucose monomers. And then those glucoses can go in the bloodstream and raise blood glucose. Now, those are how we deal with storing and releasing glucose. What about other sources of carbons? Well, lipids. Our triglycerides have carbon, hydrogen, and oxygen, but they're not in the particular proportions that we have in carbohydrates, right? It's not a 1-to-1 ratio like C6H12O6. Triglycerides are the most abundant lipid in our body, but we build those out of fatty acids. And so we can break down lipids in a process called lipolysis. You see lipo and lysis together. And we can convert the pieces into pyruvate. And then that's kind of like what we would have done in glycolysis. And then we take those pyruvates and put them into the citric acid cycle. And so we miss the parts that we got out of glycolysis, other than the pyruvates, but we can pick up from there. So we're able to produce ATPs based on these little pieces that are converted to pyruvate. So during hydrolysis, remember hydrolysis is to split with water, we're going to break a triglyceride into 1 molecule of glycerol, which is a sugar alcohol, and 3 fatty acid molecules. Now, in the cytosol of the cell, we have enzymes that are able to take that glycerol and make it pyruvate, just like glycolysis would take glucose and turn it into pyruvate. And then we can take pyruvate into the citric acid cycle, and it becomes acetyl-CoA, and we proceed from there. Now, a cell can get 120 ATP from the breakdown of 1 18-carbon fatty acid molecule. Wow, that's a lot more ATP from fatty acids than we get from glucose. 1.3 times just compared to a single 6-carbon glucose. We also make lipids. That is called lipogenesis. So you see genesis to make, lipo, what are we making? Lipids. We can use almost any of the organic molecules because lipids, amino acids, and carbohydrates can all be converted to acetyl-CoA. And then we can take glycerol from dihydroxyacetone phosphate, and it's also an intermediate product of glycolysis and gluconeogenesis. And so we have lipids that exist in non-essential fatty acids and essential fatty acids. So non-essential means we can build it. We take acetyl-CoA and we change it up, And we can build the non-essential fatty acids and steroids. So whatever we have, we can build these. Essential fatty acids means we can't build them. We don't have the enzymes. We don't have the ability to make these. So we have to get these in our diet. We have to get essential fatty acids. And the two that are most important are linoleic acid and linolenic acid. And these are found in plants. Now we store lipids. Obviously, we know where we store them. We store them in an adipose tissue, right? These are a huge energy reserve. They can give us a lot of ATP, but very slowly. And we actually tend to save these because it's difficult for water-soluble enzymes to get there. And your body will very easily make adipose tissue and fill it up. Your body's not so keen on breaking it down. We have to be able to transport and distribute it because we need lipids to make all of our membranes of our cells and of our organelles. And we need steroid hormones that are made from the same basic components to reach target cells in lots of tissues. So most lipids are not water soluble. They are hydrophobic. And so in order to keep them in the bloodstream and not let them slip through membranes, we actually have to have special transport mechanisms. We talked a little bit about this back in earlier chapters, like when we talked about the steroid-based hormones. So we have to attach them to a protein to keep them in the bloodstream. And so once we attach a lipid to a protein, we create a lipoprotein. Now there are some free fatty acids, but they're only a little bit of what is circulating. Most of our lipids are associated with proteins. Free fatty acids by themselves slip right through membranes. So it's really hard to keep them in the blood. So usually we have to bind them to albumin, which you know is the most abundant protein in your plasma. Sources of free fatty acids in your blood are those that are not used when we are creating triglycerides that come from the intestinal epithelium. So we get them in our diet and we don't use them to make triglycerides, so we have them left over. And then those that diffuse out of the lipid reserves when we're breaking down the triglycerides. So we have two sources of free fatty acids. Now, during starvation, when we cannot get glucose, our body relies on free fatty acids for creating lots of ATP. And there are cells in the liver, in the heart, in the skeletal muscle, and they can all use free fatty acids to go into the process for metabolism to create ATP. Lipoproteins, we mentioned these. These are lipid protein complexes, and they have large, not water-soluble glycerides and cholesterol. And there are 4 different groups of lipoproteins. There are chylomicrons, there are very low-density lipoproteins, or VLDLs. There are low-density lipoproteins, LDLs. Our doctors and our science has classified these as bad cholesterol. I kind of argue that, but okay. And then there are high-density lipoproteins, HDLs, which have been characterized as good cholesterol. The chylomicrons are the biggest ones. They're made by the intestinal epithelial cells that as they're breaking down fats coming in through our food. And actually, they're usually so big they can't really go into the bloodstream. And so they go into the lymph through lacteals, and then they get processed, and then eventually they'll end up in the bloodstream. So here is our little diagram here, and you can look at this at your leisure. Okay? The very low density lipoproteins transport triglycerides from the liver to muscle and adipose. The LDLs go to the bloodstream to deliver to peripheral tissues. And the HDLs are anything that is not used by the cells, and they are carried back to the liver. What about proteins? Can we use proteins as an energy source? Well, we can. However, this is the least attractive source of carbon chains. Your body uses proteins for enzymes and for structures. You actually make up to 140,000 different proteins made from the same 20 amino acids. I always joke it's kind of like Taco Bell. If you go to Taco Bell, everything on the menu is made of the same 4, 5, or 6 ingredients. You know, you got your tortilla, you got your meat, you got your cheese, and you got beans, you know, and they just put them together in all these different ways. That's how proteins work. So proteins are vital for how your body works and holding it together. We don't want to use protein as an energy source. It is us. It's mostly us. We don't want to dissolve away our own proteins to create energy to keep the rest of our bodies alive. So usually the order is we will burn glucose first, fats second, and proteins only as a desperate measure during starvation. And we have another reason why we do not want to break down proteins, because we're going to be building up a toxic byproduct called ammonia. Whenever we tear apart an amino acid, which is in the proteins, that's what proteins are made out of, we're going to create a toxic product that our body has to deal with. So in amino acid catabolism, so if we're going to break down the amino acids of proteins, we first have to get them to a form that can enter the citric acid cycle. So this conversion involves first a transamination, a deamination, and then we have to deal with the ammonium ion through the urea cycle. In order to remove the amino group, we also must have a coenzyme derivative of vitamin B6. So transamination takes the amino group off an amino acids and sticks it on a keto acid and converts that keto acid into a different amino acid. That different amino acid can leave the mitochondrion and go into the cytosol, and then it can be used to build proteins. In deamination, we're going to rip the amino group off, and what we're going to be left with is a side chain made of carbons that we can use for the citric acid cycle, but that amino group we pulled off has a toxic ammonium ion in it. So here's an example. Here's transamination. So we started with one amino acid, glutamic acid, and remember it's this side chain here that gives us the identity, right? And here's this keto acid 1. And we're going to take this amino group and we're going to move it off the amino acid and transfer it over to this keto acid, and we're going to get the amino acid tyrosine. And now we have changed that glutamic acid into keto acid 2. That one doesn't give us an ammonium ion. That one's okay. Deamination gives us the ammonium ion. So we take that glutamic acid, instead of taking this amino group off and putting it on something, we just deaminate it and we have an NH4 ammonium ion, which is toxic. So now we have to do something to get rid of this ammonium ion. So in deamination, we generate ammonium ions, especially in liver cells, which are where this is happening. Now, in the liver cells, we have enzymes that help us deal with the toxic ammonium ion by making something called urea. And it's made by something called the urea cycle. And by the time the urea cycle is finished, we create urea, and it is a fairly harmless, no longer toxic compound that is secreted in urine. That's where urine gets its name from. So this is the urea cycle, and I'm not going to ask you any details. What we do, we take 2 ammonium ions plus 1 carbon dioxide. It goes through a series of enzymes, and we end up with this molecule here, urea. Okay, so how do we use the amino acids to make ATP? So if we've run out of glucose and we've run out of fats, we are finally going to begin digesting our own proteins, often starting with our muscles. Okay? We deaminate the amino acids and we send the carbon chains to the mitochondria. But because all amino acids have varying numbers of carbon chains, they enter the citric acid cycle at different points along the cycle. Now, there are 3 reasons why we don't want to break down our proteins. First of all, they're more difficult to break down. They're more complexly structured, and enzymes have a harder time reaching the breakpoints. One of the byproducts is toxic. And finally, proteins form the most important structural and functional parts of cells, so we don't want to begin removing those just to burn it for energy. What about creating proteins? Well, kind of like we had with the fatty acids, some of the amino acids that we use to build proteins, we can create ourselves. Some of them we cannot. So there are 10 essential amino acids. 8 your body can't make it all, and 2 you don't make enough of them. So these 10 essential amino acids you must get through your diet. The non-essential amino acids we can make within our bodies. So remember, there's 20 total, so half of them we must get from our food. The other half we can convert other things into them. So here is an amination. So we have an alpha ketoglutarate. We're going to take an ammonium ion and we're going to now turn it into glutamic acid. All right, so then we're going to change gears. Nutrient requirements. So this is saying that different parts of your body require different types of enzymes to be able to perform metabolism. And the 5 main areas of metabolism in your body are the liver, that one we kind of knew, fat, maybe not have known that one, skeletal muscle, nervous tissue. So those are individual tissue areas. And then other peripheral tissues is the 5th area. So your liver, as you learned from the digestive chapter, is so busy. It has so much to do. It has its own blood portal supply that brings in all the nutrients from the digestive system. It's going to be responsible for dealing with all of those nutrients and parceling them out to the tissues and areas of the body that need it, and then keeping the energy reserves as glycogen deposits. Fat is going to be where we're going to store our triglycerides in adipocytes in the fat vacuole. And we have them all over our bodies, especially areolar tissue, mesenteries, in our bones, around the heart, and around the eyes and kidneys. Skeletal muscle has its own glycogen reserve in order to have a quick source of glucose for metabolism to keep muscle contraction going. If other nutrients are unavailable, we can actually break down the contracting proteins of muscle, things like actin and myosin, and use their amino acids as an energy source. But again, that is the last thing your body would do, and only in the process of starvation. Nervous tissue, your brain and spinal cord, and all those nerves running through your body do not maintain any reserves of carbohydrates, lipids, or proteins. And your brain can only run on glucose. So when you start running out of glucose, you will only use it in your brain, and that's when your body will switch gears and start burning fat reserves for the rest of your body. In a low glucose condition, you will become unconscious. Other peripheral tissues don't have large metabolic reserves. They can kind of go back and forth between glucose, fatty acids, and other substrates, and their preferred energy source will vary according to what the endocrine system tells them to do. Okay, so whenever you are going through your day and you are metabolizing things, you have 2 states. You have the absorptive state, where you're taking in metabolites, things that you've broken down through digestion, and that lasts about 4 hours. And then you have the postabsorptive state. In the postabsorptive state, you're supposed to have maintain blood glucose, use your internal energy reserves, and then if you need extra energy, break down lipids or amino acids, preserving the glucose for nervous tissue. So look at this. This is a typical day, and I wonder if you notice a problem here. So if you get up and you eat breakfast at 8, then for the next 4 hours, you're in the absorptive state. And then maybe you're eating between 12 and 1. Guess what? You go back to the absorptive state. Now remember, in the post-absorptive state is when you actually use the energy that you just took in. But in our American lives, here we go, we're eating again at noon or 1, and then we go back into the absorptive state. And then we're hanging on, and at 4 p.m., now we're starting to get post-absorptive between 4 and 5. And what do we do at 6? We eat again. Now that's only 3 meals. How many snacks do we eat a day? So basically, in America, we are keeping our bodies constantly in the absorptive state. We're taking in energy, and we're never using it. We're never getting to the post-absorptive state. I'm going to go to the next slide. Most people only enter the post-absorptive state when they're asleep, when their energy requirements are 30% less anyway. So I'm showing you these because there are a lot of things coming up in nutritional science right now talking about how intermittent fasting is actually good for us. And if you don't know what that is, that means going a few hours between meals. So you might decide to eat your last meal of the day at 6 p.m. And then if you don't eat till 6 a.m., that's a 12-hour post-absorptive state, right? So you get 8 hours of post-absorptive because you have to get past the 4 hours right after you've eaten. If you can extend that longer and longer, the more time you spend in the post-absorptive state, the more time you utilize the stored energy in your own body. So intermittent fasting, people say it's a fad, but it's been around for a very long time, and it seems to be the way our bodies were designed to work. Have a large meal and then wait many, many hours after processing that meal so that you get the most utilization of the energy. All right, this graphic is showing you how all components, whether they're fats, carbs, or proteins, can all be fed into the citric acid cycle and the different sub-processes. Okay? And some things are going to go off to the electron transport chain and give us ATP there. Now, when we break down lipids or amino acids, we're going to generate acetyl-CoA, which we know is the first step of the citric acid cycle. And if we have a lot of acetyl-CoA around, we can cause these things called ketone bodies to form. Now, ketone body is a fatty acid metabolism organic product, and it dissociates in solution and it releases a hydrogen ion. There are three types. There's acetoacetate, acetone, and beta-hydroxybutyrate. These are not able to be catabolized by liver, and instead your peripheral cells will absorb them, and then they can make them back into acetyl-CoA for citric acid cycle. So whenever you fast, you produce these in a process called ketosis, when you're relying on these ketone bodies. This is not a bad thing. The only time it's a bad thing is if you either have type 1 diabetes and you produce an excess amount of these, or you are starving in starvation, then you might have a problem with these excess hydrogen ions building up and lowering your blood pH, and then you can get something called ketoacidosis. But this does not happen if you're just doing a couple of days of fasting. This is prolonged starvation. So a lot of people are afraid of the ketogenic diet because they say, oh my gosh, you're going to die from ketoacidosis. That is absolutely not true if you are following the diet regularly, and you can even include a couple of days of fasting. You will be fine. You will not enter this state as long as you are not a type 1 diabetic who's going to have high blood glucose and high ketone bodies at the same time. Okay, shifting gears again. Now the chapter is just talking about some general definitions. So since we need these building materials to maintain homeostasis, we're going to define a few terms like nutrition. Nutrition is the nutrients that you are able to get out of your food so that you can build up and perform catabolism and anabolism. A balanced diet has all the ingredients your body needs to make all of the essential fatty acids and essential amino acids. Malnutrition is when you're missing one or more ingredient for making these proteins, carbs, or fats that you need in your body. Now, when it comes to proteins, we have complete and incomplete proteins. A complete protein within its structure has all of the essential amino acids. Remember, there are 10 that are essential. So things like meats of beef, fish, chicken, turkey, eggs, and milk have in their makeup all 10 of the essential amino acids that our bodies need. So they're called complete proteins, and you can eat them and live on them alone. Incomplete proteins means that we're missing one or more of the essential ones. Now, plants are often incomplete proteins. So we have to combine one incomplete set from one plant with an incomplete set from another plant if you're going to only eat plants, and that will give you all of the 10 essential amino acids. And a lot of cultures have learned these combinations. So beans and rice. It's a very popular combination because the rice provides some of the essential amino acids, the beans provides the missing ones. So if you eat them together, you have a complete protein. There are a lot of combinations like this, like chickpeas and rice, and all sorts of things like that. Minerals are inorganic materials that are going to be released through breaking down of electrolytes, salts, and we need them for things like our sodium-potassium pump. We need them for physiological processes, and some of them are needed to be cofactors for enzymes. Bulk minerals that you need: sodium, potassium, chloride, calcium, phosphorus, and magnesium. You need a lot of those every day. Trace ones, you just need a little bit of iron, zinc, copper, manganese, cobalt, selenium, and chromium. And your body is able to store some of these in reserves. Vitamins are going to work as coenzymes. Sometimes they're needed to help an enzyme work. Sometimes they're needed to help maintain the shape. And vitamins come in two forms: fat soluble and water soluble. So one loves lipids and oils and one loves water. The fat soluble vitamins are A, D, E, and K. Now, when you go to the supplement section of your grocery store or your drugstore, you'll notice that these come in oil caplets because they can only be dissolved in lipids. Okay? You need all of these vitamins. Vitamin A is for eyesight, vitamin D is for calcium absorption, vitamin E is for immune function, and vitamin K is for clotting. So we need all of these fat-soluble vitamins. Vitamin A also maintains epithelia. Vitamin E helps prevent the breakdown of vitamin A and fatty acids. And as I mentioned, vitamin K is essential for clotting. So you can hold pretty large reserves of fat-soluble vitamins. So even if you're missing those for a day or two, you should have enough stored in your body. Hypovitaminosis is vitamin deficiency. And because you can store them in your body, this usually doesn't happen with the fat-soluble vitamins. Hypervitaminosis is when you have too much and you've exceeded your body's ability to use or excrete a particular vitamin. That might happen with the fat-soluble vitamins. Water-soluble vitamins are usually coenzymes. And you get rid of any extras that you take through your urine. So you don't have to worry about hypervitaminosis with water-soluble vitamins. You might have to worry about hypovitaminosis because you also don't keep reserves of these in your body. Now, we rely on our intestinal bacteria to make some of our vitamins and get them out of our food for us. 5 of the 9 water-soluble vitamins are contributed to by the bacteria in our guts, and including the fat-soluble vitamin K as a 6th one. We can absorb all the water-soluble vitamins except B12 through our intestines. But B12 is so big, we have to bind it to an intrinsic factor before we can absorb it. Metabolic rate is how much energy do you expend daily through activities and metabolism. And energy gains and losses, of course, are building and breaking of chemical bonds. And we're going to have body heat produced as some of a byproduct that's going to lose some of the energy. So to measure energy, the definition of the amount of energy required to raise the temperature of 1 gallon of water 1 degree Celsius is a calorie. Now, this is going to make you feel really bad because the calorie that you see on your wrappers, on your food, isn't really that calorie. It's actually the second definition. Energy required to raise the temperature of a kilogram of water 1 degree Celsius is a kilocalorie. And that's the one they put on our wrappers. So if you want to look at it as like 1 gram of water, 1 degree Celsius, all your calorie counts on your food, let's say a serving is 150 calories. It's actually 150,000 of these. So this one is written with a capital C. This one is written with a lowercase c. And I'm sorry, I think I said 1 gallon. That's 1 gram. So we can figure this out by actually using a device called a calorimeter. And by using a calorimeter, they've come up with these calculations that per gram of lipid, we get 9.46 kilocalories. Per gram of carb, we get 4.18 kilocalories. And per gram of protein, we get 4.32. So about 4 kilocalories per gram for carbs and protein. And lipids are much richer. We get about twice that, 9.5 kilocalories per gram. Your metabolic rate is how many calories per hour, calories per day, or calories per unit body weight per day that you use. And of course, what you do during the day affects this. Your exercise level. Your age affects it. The older you get, the less metabolism you have going on. Your sex, your hormones, and your climate. Okay? This is showing you different activities and how many calories per hour they burn. Your basal metabolic rate is what your body burns just at rest. Okay, and actually having a high basal metabolic rate is actually a good thing. It means you're burning more calories even when you're not doing anything. You can read these last few slides. Obesity is defined as having a body weight more than 20% above your ideal weight. And as we all know, America has a huge number of technically obese people, probably most of us. And it has been associated with heart disease, cancer, and diabetes. Okay, guys, that's the end of this chapter. Hi. So we will start the next chapter in the next video.